Today's lesson is about enzymes. First, we need to talk about metabolism. Metabolism is the sum of all the chemical reactions that happen inside our bodies. So all of the chemical reactions of life, and those include forming bonds and breaking bonds. Those are all the chemical reactions that we talk about. And we've talked already about dehydration synthesis and how we form bonds. And hydrolysis is how we break bonds within molecules. We're going to add one more vocab term to this. When bonds are formed between molecules, that is considered an anabolic reaction. And when bonds are broken, that is a catabolic reaction. Chemical reactions can either release energy or require an input of energy. Chemical reactions that release energy are considered to be exergonic. Think of it that energy is released, energy exits the molecule, or the reaction. And hydrolysis is an example of an exergonic reaction. When we break apart, when we break bonds, energy is released. And here over on the side, you can see in the graph here that we start with our reactant molecules. We have some energy, but the products are way down here for energy, and that means we've had a release of energy. So the amount of energy from this line right here going down means that energy was released. And then an endergonic reaction is the opposite. It requires an input of energy, so that means that our products are going to be way up here and that we need a lot of energy to make that reaction happen. It needs to be supplied. Think of endergonic as enter. Energy enters the reaction. And again, dehydration synthesis, where we build molecules, right? Building things requires energy. So those would be endergonic. Organisms require energy to live, right? And we can't continue to take in an ongoing supply of energy with the food that we eat. So what happens is that in our bodies, chemical reactions are coupled. So we couple exergonic reactions where energy is released with endergonic reactions that need energy. So the energy is conserved. That one releases energy, exergonic. The energy goes over to the endergonic reaction so that these molecules can be built. So the energy released from this reaction here fuels the other reaction. Activation energy is the amount of energy that's required to get the reaction going, to break down these molecules. That is the initial input of energy that's needed to break down these large molecules. And a catalyst is something that reduces the amount of activation energy. It helps facilitate the reaction, so it reduces the amount of energy that's needed. And in biology, we call those catalysts, I have a typo there, enzymes. Enzymes are biological catalysts. And you can see here in this graph, this is the amount of energy before we added enzymes, before it was catalyzed, and the new amount of energy. So this is the new activation energy, whereas before, without a catalyst, the activation energy was much greater. Enzymes, there are a variety of different molecules that count as enzymes. They are mostly proteins. Proteins are the majority of enzymes, and sometimes RNA can act as an enzyme. We will talk about that later in the protein synthesis section of this. Enzymes facilitate chemical reactions. They increase the rate of the reaction without being consumed. That means that the enzyme doesn't go away. It's still there, so it can be used for the next time that reaction has to happen. They reduce activation energy. They don't change the amount of energy released for the actual reaction itself, so they don't change the amount of free energy. They are required for most biological reactions. They are highly specific. We will talk about how specific they are. We have talked about that before, with the lock and key system that enzymes have. We're going to expand on that some more. And they control all of the chemical reactions. Some vocab that you need to know is the substrate. That is what the enzyme binds to. That is the molecule that the enzyme is going to act on. The products are the end results of what happened. And then the active site is on the actual enzyme. It is the location that is specifically fit for the substrate to bind to. Enzymes, again, are very specific. Each enzyme works with a specific substrate. There is a very specific chemical fit that occurs between the active site and the substrate. That fit occurs due to hydrogen bonds and ionic bonds. They are not consumed in the reaction, so a single enzyme molecule can catalyze thousands or more reactions per second. So the enzymes are unaffected by the reaction. But enzymes can be affected by cellular conditions. Any condition that affects the protein structure, right? Because an enzyme is a protein. So things like temperature, pH, salinity, those are all going to affect the structure, right? The secondary, the tertiary, and the quaternary structures of proteins. And if they have a specific shape that they have to be, then this is going to affect them significantly and their ability to work. Naming conventions. Enzymes are generally named for the reaction that they catalyze. So some examples here. Sucrase is the enzyme that breaks down sucrose sugar. A protease breaks down proteins. Lipases break down lipids. DNA polymerase builds DNA. So usually the names are connected to the substrate that they act upon. The lock and key model is a simplistic model of enzyme action. The substrate fits into the three-dimensional location of the active site. That molecule would fit in there. There would be hydrogen bonds that occur between the substrate and the enzyme in this active site, and it acts like a lock and key where the shapes have to match, or else the enzyme is not going to catalyze that particular substrate. A little more detailed is the induced fit model. Basically, you can think of this as the enzyme giving the substrate a hug. The active site of the enzyme is here if you look, and once the substrate gets into that active site, the enzyme itself kind of hugs around it to ensure that that substrate is not going to become detached or leave that active site until the reaction has actually occurred. So what we call this is that the enzyme undergoes a shape change, and in science we call that a conformational change. Anytime we talk about a shape change, it is a conformational change, and it's going to bring the chemical groups in better position to make the reaction happen.